Good afternoon, everybody, and maybe morning still to some people. My name is Chris Griffiths, and with Eli Sprecher, Professor Sprecher from Israel, we'll be presenting the today's ESDR Kitchen. I was going to give you a little bit of background to any of you who have not been able to tune in to us before. So ESDR Kitchen is something that the ESDR set up when we, very soon after we realized that we would not be running any face-to-face meetings because of COVID over this year and maybe into the beginning of next year. And it's a way of maintaining our connections with our ESDR membership and further afield, and also providing scientific and educational content. We have four themes to the kitchen. The recipe book, which is really research techniques explained. Freshly Baked, where we'll be talking about the latest publications in high impact journals which are relevant to dermatology, and Molecular Cuisine, which is a potted life history or life story of one of our senior researchers from around the world as to how he or she has made the discoveries that have marked their career. But today's theme is returning to sweet and sour, which is where we have two eminent scientists will discuss a hot topic in dermatology and dermatology research. And if we could go to the next slide. So today I'm delighted that we've got two people from really about as far away as we can find, actually. Rich Gallo from the United States, who I'll introduce in a moment, and also Scott Byrne from Australia. And they're going to discuss vitamin D and inflammation. And at this juncture, I should say that today's kitchen and subsequent 5 are supported by Amgen. So the first presentation is actually pre-recorded by Rich Gallo because he is in San Diego and it's only about 4 o'clock in the morning their time. So Rich Gallo is well known to all of you, I'm certain, as a very eminent skin scientist. He's professor and chairman of the Department of Dermatology at the University of California, San Diego, and has made very important discoveries in the world of dermatology, particularly the role of antimicrobial peptides. But today he's going to be setting the scene for vitamin D and inflammation. And those of you who can increase your volume, I suggest you increase the volume on your computers because the recording is a little bit quiet. So Rich Gallo.
Hello, I'm Rich Gallo from the University of California, San Diego. And I want to thank the ESDR for inviting me to be part of their ESDR kitchen. I'll be presenting the position that vitamin D is beneficial to immunity and human health. And let me get started since we don't have very much time here. This is a complicated subject. But since I'm starting off, I thought it would be necessary to begin with some basics. So first, it's important to recognize that there are 2 main sources of vitamin D nutritionally. One is orally in terms of what can be dietary intake, and the other is through the well-known and popularized photoconversion of 25-hydroxy cholesterol to first to pre-vitamin D and then the vitamin D3. Importantly, vitamin D itself is not biologically active. Hydroxylation of vitamin D is necessary to convert it to active forms. The liver is a but not only source of 25-hydroxylation to make 25-hydroxy vitamin D. And then further conversion by a 1-hydroxylase activates the 25-hydroxy vitamin D to 1,25-hydroxy. And it's generally believed that 1,25-hydroxy vitamin D is the most biologically active. The main controversy, or one of many controversies around vitamin D status, has been what is adequate. Now, several years ago, I was fortunate to take part in the U.S. Institute of Medicine analysis of what's adequate for vitamin D supplementation and intake in the normal population of North America. And the end of about a year analysis by about 25 independent experts concluded that based on the available evidence, the Institute of Medicine felt that a serum 25-hydroxy vitamin D level of about 20 nanograms per mil was adequate for 97.5% of the healthy population. Shortly after that, a couple other societies somewhat complicated the story. The American Endocrine Society felt that a higher serum vitamin D level was necessary, and the Vitamin D Council went even further to say almost twice the minimum of 40 nanograms per mil is adequate. And as you can see, there are various, these different groups have various ranges on what can be sufficient, can have potential adverse effects, and what is insufficient and what is highly deficient. I won't get more into this controversy other than to recognize it, to mention that the members of the IOM committee did not have the same kinds of conflicts of interests with for-profit groups that make their profit based on either selling vitamins or testing for vitamin D. So there may have been some implicit bias in this, but of course, as in all of medicine, there's always controversy. Another important thing to recognize is that there's not a linear relationship between that 25-hydroxy vitamin D level and how much vitamin D you take. This is a study that was a Cashman study that was very nice, published back in 2008, that looked at the amount of intake of many individuals when they started at different vitamin 25-hydroxy D levels in the serum. And what could be clearly seen from this study is that if you start off at inadequate or insufficient levels, increasing your nutritional intake of vitamin D clearly will raise your 25-hydroxy vitamin D level a significant amount. However, in those population that already have sufficient vitamin D levels between 50 and 100 nanomoles per liter, that population, when you start taking more and more vitamin D, it doesn't really do much. Yes, you can drive your vitamin D levels to toxic levels if you take a lot, but the body does a lot to regulate. So adequate nutritional intake is important, but additional supplementation within reason may do very little at all to the serum hydroxyvitamin D levels. Another thing to comment on is what influences the serum vitamin D levels is not simply only due to, say, sunlight exposure or nutritional intake. This is a very interesting and important study, I felt, that was published again back in 2009. It was based on a twin study. It looked at the vitamin serum 25-hydroxy D levels in a number of twins during the summer months and during the winter season, and as seen in many other studies with greater sunlight exposure, vitamin D status increases. However, importantly, the variance in this was tremendous, and even twins with the same UV exposure could show quite significant differences in their serum 25-hydroxy vitamin D levels. So in addition to obvious sources for variance between populations such as intake or UV exposure, genetics has a big part of this, and this study concluded that up to 50% of the variation is purely due to other modifying genes that were are still yet not well understood. So the molecular biology of 25-hydroxy vitamin D, as I mentioned, that 25-hydroxy D is hydroxylated to become 125-hydroxy D, which is the most active but not the only active form of vitamin D that can bind the vitamin D receptor and has important transcriptional effects on many different types of genes. These observations are non-controversial. The in vitro studies have clearly shown that vitamin D and these metabolites are important for gene transcription. There are also a number of non-genomic effects that have been reported and validated in vitro. I won't get into this, but it's clearly 125-hydroxy vitamin D is an incredibly important molecule for regulation of cell behaviors. We got into this field some years ago when Jürgen Schauber in my lab, and then later Beta Mulheisen, and also in collaboration with Robert Modelin at UCLA, observed that the skin and the immune system have important roles in vitamin D metabolism. And in fact, keratinocytes themselves, we showed, express that 1-hydroxylase as well as the 25-hydroxylase. So there can be local activation of vitamin D. And the model we proposed at the time is that following injury and the release of factors such as interferons and TGF-beta, these immune signaling molecules can activate the keratinocytes' capacity to express 1-alpha hydroxylase, and that then converted local 25-hydroxy vitamin D from serum to this transiently and highly potent 125-hydroxy D source. That 125D then could act again back on keratinocytes, increase the transcription and expression of innate and microbial recognition molecules such as toll-like receptor 2 or CD14. It also had direct effects on the expression of cathelicidin antimicrobial peptide. And this is quite a potent effect. And indeed, in mice that were severely vitamin D deficient, if you supplemented them and gave them more normal 25-hydroxy vitamin D levels, that could improve their resistance to infection. And that was cathelicidin dependent. However, in the human population, the data are less clear. What I believe to be the strongest data about the role of vitamin D has to do with bone health. And of course, we all know that the disease rickets, which luckily we do not see very often in the industrialized society today, is very rare due to now having adequate vitamin D sources. And there have been a number of studies looking at many randomized control trials, both retrospectively and prospectively, that have suggested that bone and muscle health can be improved in people who are deficient before they start vitamin D supplementation, and with supplementation, their bone health improves. What's less clear, however, are the direct immunological effects of vitamin D supplementation. Again, there's many associated studies that have indicated that vitamin D status is relevant to immune function, perhaps the most obvious to the population in flu. And since vitamin D levels decrease in the winter and in the fall, and flu occurs more frequently in those seasons, there is an inclination to believe that the vitamin D serum levels are somehow preventing flu. Indeed, this study stratified the incidence of upper respiratory tract infections within these months and suggested that those individuals with the very, very lowest, again, this is now low, insufficient levels of 25-hydroxy D, but those low serum hydroxy D levels had the highest incidence of flus. And then there are many studies which are listed here. I'll refer you to this recent review article, which argued strongly for the benefits of vitamin D in immunology, but it's summarized some of the papers looking at envelope viruses and the association between different envelope viruses like dengue fever and HIV, and how vitamin D supplementation might decrease aspects of inflammation. And then, of course, now with the COVID pandemic occurring, we're seeing quite a lot of renewed interest in vitamin D. There's a recent letter that stated vitamin D mortality increases with low northerly latitude, suggesting a link between ultraviolet light and vitamin D. And this was a study that was recently in JAMA Open Network that shows that the vitamin D level before the COVID test, that more patients seem to test positive if their vitamin D levels were low. Now, I don't argue with these data, and since I'm supposed to be taking the position that vitamin D is very important for human health, I hope I've convinced you that biologically, in terms of the cell biology and the immunology of this, that there's clear roles for vitamin D in human health. However, the in vivo observations suggest that there may be a number of confounding variables which affect conclusions. There is quite a bit of metabolic machinery which is dedicated to maintaining normal vitamin D levels. Becoming vitamin D deficient is perhaps indication of other diseases, and those other diseases in the vitamin D deficient individual may be what's contributing to the association between infections and vitamin D. So with that, let me just end by concluding that we really have a lot more to learn about the immune benefits of vitamin D, but please be aware of many false claims in the news that are not substantiated by strong science. And I hope that careful scientific investigation will improve our understanding of this. So with that, I want to thank you. I look forward to hearing the next presentation, which should go into more of the difficulties in association between health and vitamin D status. And should you have any comments or questions for me, you can always be reached by email. Hopefully we'll all be together soon once this pandemic has ended. So best wishes.
Well, thank you so much, Rich. And I think that really worked very well. That's a beautiful scene setting. So I'm now going to introduce our second speaker, who's Scott Byrne. He's an immunologist and associate professor at the University of Sydney. He leads the cellular photoimmunology group at the Westmead Institute of Medical Research. And he's very well known in the field of photobiology and won a number of important awards, including being the Finson Lecturer at the International Union of Photobiology annual meeting just recently. So Scott, delighted to welcome you, and thank you for staying up so late over there in Sydney. And we're looking forward to your lecture on sunlight.
Thanks, Chris. And thank you. I hope I'm not too loud for you all after that presentation. You might want to turn down your volumes a bit. It's a great privilege to be able to speak to you. I really wish I was in Europe. I enjoy coming to Europe, particularly at this time of year, when it's just starting to get really hot here in Australia. So let me get started and perhaps start by saying that Rich and I are in furious agreement with each other. And we decided we wouldn't do a debate necessarily because we were agreeing with each other a lot. So yes, the vitamin D pathway is involved in inflammation. What I'm planning on talking about to you today is highlighting some of the deficiencies around why we think suppressing inflammation by boosting vitamin D levels will work, and why many of the oral supplementation studies haven't worked. And I'll try and explain why I think some of that might be the case. And at the end, I'll come back to thinking about some of the ways which we can take advantage of the anti-inflammatory properties of sunlight and vitamin D in a safe and effective manner. So first off, what evidence is there that the vitamin D pathway is anti-inflammatory? Well, vitamin D has been known to be immune suppressive for quite some time. And when I say immune suppressive, I'm talking about suppression of the cell-mediated or adaptive immune responses. So vitamin D, for example, in mice can enhance the suppressive activity of regulatory T cells. In humans, topical applications have been used quite widely now for treating a range of inflammatory skin diseases, and they can suppress skin immunity in humans. And Peter Wolf's group has shown over many years of dedicated research that various vitamin D analogues can be used to protect from inflammatory skin diseases like polymorphic light eruption. So if vitamin D deficiency is associated with so many inflammatory diseases, why then doesn't artificially boosting vitamin D, particularly through supplementation, work more readily and more effectively? Well, perhaps one explanation is that inflammation itself increases the enzymes that break down the active form of vitamin D. And in mice, this is a lovely study done by Professor Pruhart here in Australia, where they demonstrated that when mice were fed high vitamin D diets and then given an inflammatory disease, and this inflammatory disease happens to be an inflammatory bowel disease model of ulcerative colitis. When they induced inflammation, the vitamin D levels in those mice, despite being on very high doses of vitamin D, significantly fell to almost negligible levels. And so this perhaps gives us a hint as to why we need to control inflammation before we try to boost with vitamin D. In humans with inflammatory bowel disease, we see similar failures with vitamin D supplementation. This is another Australian study, this time done in Victoria. And this study, published in Clinical Nutrition just a couple of years ago, showed that a specified oral vitamin D regimen did not alter any objective measures of intestinal or systemic inflammation, despite boosting vitamin D levels as expected. This slide from Rich, just as to segue into and remind you of one of the more efficient ways in which vitamin D can be made, and as Rich has shown, can be made entirely in the skin, and that's through exposure to the ultraviolet radiation contained in sunlight. And here in Australia, sunlight and vitamin D is associated with human disease, and a latitude gradient exists for a number of these diseases. Perhaps the most well-known here in Australia is melanoma and skin cancer. And certainly Queensland, who today had a UV index of 13, despite only being in mid-spring, skin cancer is still a massive problem in Australia. But there are other latitude gradient effects that exist, but they're reciprocated. In other words, more UV is associated with less of these diseases, including asthma, cardiovascular disease, colitis and colorectal cancer, as well as autoimmune diseases. Perhaps the most well-studied and well appreciated is that of the association between sunlight exposure and multiple sclerosis, which is an autoimmune disease that affects the central nervous system. A studies done about 30 years ago now showed that if you're born and bred in Tasmania, which is very southern latitude, you're about 6 times more likely to develop multiple sclerosis than if you're born and bred in far north Queensland. This latitude gradient is replicated across the globe in Europe, America, and in South America as well. So this begs the question, could UV-induced vitamin D explain this protective effect against inflammation in the CNS? Well, we know that high circulating levels of vitamin D is associated with lower risk of MS. Increased MS disability is associated with lower serum levels, and giving vitamin D to mice prevents an MS-like disease in mice. But perhaps, as Rich alluded to, some of the most compelling evidence that vitamin D is involved in these types of inflammation is genetic studies. And a GWAS done by Ansgene about 10 years ago now clearly demonstrated a role for CYP27B1, the gene which gives rise to the 1-alpha hydroxylase, which converts the inactive form of vitamin D into the active form. And again, I borrowed this figure from Rich's slide to show you where this gene fits within that pathway. Unfortunately, vitamin D supplementation in this disease does not seem to be sufficient. Many studies have shown that there is no clinical benefit from giving vitamin D to MS patients. And even in some of the mouse studies, it's shown that the effects of UV are independent and not due to vitamin D. And in fact, in some cases, vitamin D deficiency may even diminish EAE severity. In other words, exactly the opposite to what one might expect. In more recent human trials of vitamin D supplementation, here's a study from Australia again showing in neurology, showing that there was no therapeutic advantage in relapsing remitting MS for high dose vitamin D2 over low dose vitamin D2. But that's really putting a very positive spin on what really was perhaps deep down in the paper was this conclusion: those on high dose actually had a higher expanded disability status scale and were significantly more likely to relapse than those on low dose. So in fact, high doses in this particular trial were led to worse outcomes for patients than lower doses. This trial came out this year. It's the highly anticipated eVitamus trial, the European trial of vitamin D supplementation. And for those who aren't familiar with this trial, it was a multi-center randomized and stratified active controlled double-blinded phase 2 trial that involved 2 doses of vitamin D, a high dose or a low dose, and all participants were on a standard disease modifying therapy, in this case interferon beta, starting 3 months prior and throughout the study. The outcomes, I think it would be fair to say that they were very disappointing. After starting recruitment, the study was terminated early for inexplicable reasons. 41 patients completed the study, and there were 41 adverse events. And there were more adverse events in the high dose vitamin D arm. And curiously, most of those, the more common adverse events were respiratory infections and musculoskeletal complaints. And most importantly, there was no difference between high and low dose vitamin D on any clinical or MRI parameters. So this has led many people, including ourselves and Peru Heart and others, to investigate whether sun itself, rather than vitamin D, is associated with multiple sclerosis. And I've just sort of captured one of the highlights from this particular study, which was very well conducted, a Californian study, that lifetime sun exposure appears to reduce the risk of MS regardless of race or ethnicity. In contrast, serum vitamin D levels are not associated with MS risk in Blacks or Hispanics. And our findings challenge the biological plausibility of vitamin D deficiency as causal for MS. And I think that's a really important take-home message. So what about other diseases and other all-cause mortality? And some of the best work's been coming out of Europe on this particular question. And the risk of all-cause mortality is reduced in women who went on a sunbathing vacation a year. However, and this is really important, solarium use was actually associated with an increased risk of all-cause mortality, and the authors couldn't explain why that might be. Most of the excess mortality due to insufficient amounts of sun is due to increased risk of death from cardiovascular disease. And we've known about the association between sunlight and cardiovascular disease for some time now. This is a very old study, almost 30 years ago, in the Lancet, showing that it was reasonable to conclude that the further one lives away from the equator, the greater one's risk of ischemic heart disease. And vitamin D levels do correlate with a patient's susceptibility to cardiovascular disease. However, and as Richard Weller has well shown, vitamin D supplements have no effect on reducing cardiovascular disease outcomes in humans. And in fact, in this systematic review published not that long ago now, a very well conducted systematic review, 34 studies, including over 2,800 individuals with mean vitamin D concentrations of lower than 50 nanomoles at baseline, did not show any better results from supplementation. In this case, even for musculoskeletal diseases where we perhaps have the best evidence that vitamin D has a benefit, the authors came to the conclusion that vitamin D supplementation does not prevent fractures or falls or have any clinically meaningful effects on bone mineral density. So this really is quite surprising. And there were no differences between the effects of higher and lower doses of vitamin D. And the authors concluded that there is little justification to use vitamin D supplements to maintain or improve musculoskeletal health. And this has led leaders in the field, and many of you recognize the authors on this very recent review, to come to this startling conclusion that insufficient sun exposure, not vitamin D itself, may be responsible for over 340,000 deaths in the United States and almost 500,000 deaths in Europe each year. And that vitamin D has long been considered the principal mediator of beneficial effects of sun exposure. However, oral supplementation has not been convincingly shown to prevent any of these conditions. And therefore, and we must conclude that serum vitamin D as an indicator of status may be a proxy for and not a mediator of beneficial effects of sun exposure. So am I advocating for more sun exposure? No, I'm not. I think that would be a really quite a dangerous thing to do, particularly as for an Australian, and particularly to a dermatology audience. We're not asking people to go and sunbake and get themselves into the frying pan, because we know the dangers of sunlight exposure. They're well described. We get sunburn, we get photoaging, and we get skin cancers that are deadly. And luckily we do have new pharmacological tools at our disposal, particularly immunotherapy at the moment, for dealing with these problems. But at the moment, we still need to do much more work in understanding the effects of sunlight on the immune system. So this has led me to ask the question, is there another way? And Prue Hart has been leading the charge here by demonstrating that artificial UV radiation, many of people in this audience will use phototherapy in their clinics to try and exert health benefits from UV in other cases other than skin. And for those of you who aren't familiar with this really groundbreaking trial, clinical isolated syndrome is the first clinical presentation of a disease that shows characteristics that are consistent with MS. But it's yet to fulfill the criteria and dissemination in time and space of MS. And many CIS patients will go on to develop MS. And so the time immediately following a diagnosis of CIS could be the best chance that we have to intervene with UV. And that's exactly what Prue Hart has done. This is a photo of us at a recent ESP conference, which we love to attend. And in this trial, Prue and the dermatology colleagues took CIS patients, they randomized them to receive narrowband UVB radiation to their full bodies on 3 occasions over 8 weeks. All the patients were supplemented with vitamin D to rule out any effect of vitamin D deficiency in this case. And the primary outcome I'm going to show to you today was the development of a new demyelinating event. In other words, progression to full-blown MS. This is the results from this study, which you can find that are now published. And while the days from CIS diagnosis to MS did not reach significance, there was a substantial effect on the ability of UV to prevent the conversion of CIS patients to MS, thus showing and demonstrating an effect of UV independent of vitamin D. But the other way that you could do this is you could target the cells in the skin directly perhaps, or even in the parts of the immune system that are responsible and responding to vitamin D. And this is work done at the Westman Institute by David Booth and colleagues, showing that there are specific autoimmune risk genes that are responsive to vitamin D stimulation. But not all cells of the immune system respond to vitamin D in the same way. And what this figure from the paper shows is that dendritic cells, those cells which are responsible for starting the immune system, are particularly sensitive to vitamin D stimulation. So maybe there's a way in which we can use vitamin D to target these cells more specifically and more effectively. And so in conclusion, I'm advocating for harnessing the power of solar ultraviolet radiation, but safely. We won't be changing the safe sun message in Australia anytime soon. That would be premature. But we do need to prevent and treat these diseases safely without risking an increase in UV-induced skin cancers. And I think there's a way in which we can manipulate or replicate or enhance the effect of UV and potentially enhance the effect of vitamin D. But we need more work. We need to understand what is a healthy amount of sunlight exposure. Is it chronic low or is it intermittent high? Is it UVA, UVB, visible infrared, or a combination of these things? And when is the best time in life to get that sunlight exposure? Is it as a neonate, child, adolescent, adult, or should it be ongoing throughout life? And I think that's where I'll leave it and send back to my hosts for the discussion in this area.
Thank you very much, Scott and Rich. I think that I see that you join us. So this was really a brilliant lecture, and I think it really set the stage for many more questions in the near future. Because what we realize just hearing your lecture is that we actually have much more to learn than about this topic, but there is a lot also to harness from that in terms of future treatment. So we have a few questions. One question is, does ingested vitamin D has the same effect on inflammation in general and in the skin specifically as skin-produced vitamin D? Scott?
Me or Rich? Is that who's going to answer that one? Rich, are you there?
I don't know if...
I'm here, but you know, that is controversial, and to my knowledge, there's not clear evidence to support a difference in terms of the vitamin D outcome based on the source.
Certainly, though, there's good evidence that topical applications work in some inflammatory skin diseases, right?
The topical vitamin D is a form that is already activated, so that'll be a analog to 125 hydroxy vitamin D, not vitamin D3 or vitamin.
So we have another question here. So the question is whether to, so we could actually give phototherapy to skin areas that are usually skin protected, such as the buttock, while protecting the face and arms, and this could actually, on the one hand, maybe provide the beneficial effect of that therapy, if you understand well the question, Scott.
Yeah, possibly. I think the surface area for phototherapy is crucial for it to exert systemic effects. Certainly, you know, local application of phototherapy for skin diseases like psoriasis, for example, and contact dermatitis have very localized effects. I'd be a bit concerned about exposing skin like buttocks like that to higher doses in case that there would be, if unprotected sites might be more prone to having damage associated with them, depending on the type of phototherapy used, of course.
And then maybe another question. I think that just listening to what you were speaking of regarding the potential beneficial effect of sun exposure on CIS or MS. So I guess the $1 million question is, what is actually mediating the beneficial effect of sun exposure? I mean, any speculation regarding what vitamin D apparently is not
responsible for? Yeah, so our animal models tell us that there are possibly 2 or 3 mechanisms by which UV itself might be having an effect on diseases like MS. One is by targeting Langerhans cells in the epidermis to drive regulatory T cells. The other is, and this is work we published just a few years ago, showing that activation of regulatory B cells might also play a particularly important role. And in fact, some of the subsequent work we've done looking at the immune cells in the patients who were exposed to the phototherapy shows that there is a dramatic impact on B cells in particular. Now, exactly how the B cells are affected by ultraviolet radiation is not yet clear. But what is clear is that there is a profound change in the types of B cells that are in the circulation following exposure to phototherapy in these CIS patients.
Okay, and then I think we have time just for one last question, which is, does ingested vitamin D has any effect on skin inflammation? Any evidence for that?
Rich, do you want to answer that one?
Well, again, it's always important to remember what's the vitamin D status before it's ingested. So I would suggest that if you are within the normal range of 25 hydroxy vitamin D, ingestion within the subtoxic levels, does it have good evidence of having an effect?
Yeah. Okay. So I think that we have, unfortunately, to end here, but we will refer any remaining questions to our speakers, and they will hopefully get back to you with answers. So I'd like once again to thank you very much, Scott and Rich, for your fantastic lectures, and to you and all the rest of the audience, I invite you to our next Kitchen Live events on November 11, in the frame of the recipe book session. We have OK Otten from San Diego will introduce us to the CRISPR genetic screen, a very timely topic after the last Nobel Prize presentation. And then 2 weeks later, Kathy Green from Chicago, she will speak on a topic to which she has dedicated most of her career, epidermal cadherins. This will be on the 25th of November, and that's a new edition of our molecular cuisine section. So please be sure to be there. And in the meantime, I wish you all a very nice day, a relaxing night, Scott and Rich, and to all of you, stay safe. Goodbye.
Thank you. Bye-bye, everyone.